Good morning. I'm Dr. Lalit Dar. I look after the virology lab at AIIMS. Today we will have our first class on Introduction to Virology. I don't know whether any virus needs any introduction anymore. Since the beginning of this year, we have all been grappling with COVID, and I think now we've probably had an overdose of virology without my ever having had to teach it. In any case, we'll talk about the various groups of viruses today. The intent is just to familiarize you with the properties of viruses, with the classification, with their structure, and a little bit about their replication. And at the end of it, you should also be able to know about the kind of diseases that they cause. So it's an overview kind of thing, but hopefully we'll try and complete that first, and then Dr. Ashish Chaudhary, who's supposed to take the next class, is unable to do so today, so I will be covering that, and we'll probably have a 5-minute break in between, and then I'll come back and tell you about the laboratory diagnosis of viruses. So we start with what are the properties of viruses and how they differ from other pathogens like bacteria. Now, in this table, we have a comparative sort of presentation of what are the properties of viruses, bacteria, fungi, and parasites. The key thing is that they are not cells themselves. They are not really like cells. So they don't have a lot of the things that are required to function or to replicate themselves. So they need to be dependent on a host cell for that purpose. And that is one of the major differences between viruses and other pathogens. The next important difference is that they are either have DNA or RNA. Their genome is comprised of either DNA or RNA, not both. Unlike other pathogens and humans who have both DNA and RNA, the viruses have only one of these types of nucleic acids encoded. They don't have any organelles, so they don't have a nucleus, they don't have mitochondria, they don't have ribosomes, and all these properties also make them different. And as far as the structure goes, they don't have anything really rigid outside. Either they have a protein capsid, which happens in non-enveloped viruses, which means that they have a tightly packed nucleic acid inside what is called a capsid, which is basically tightly interlinked proteins, which maintain the integrity of the virus. Or some of these have envelopes outside, and these envelopes are basically lipids. And they have they have some kind of glycoproteins emerging out of that lipid envelope, and so these are the two types of coverings that you will see. Where the envelope is there, then inside that the capsid is present. And finally, they are as I said, they are obligate intracellular pathogens, and therefore their replication is dependent on the cell. So to summarize, there is one more property, that is that of size. They are too small to be visible under a light microscope. And the resolution of a light microscope, if you recall a little bit of your physics, is about half the wavelength. The resolution is the ability to distinguish between two close points under a microscope. And for that, it works out to about half lambda by 2, which is about half the wavelength of visible light if you're using a light microscope, and the wavelength of visible light is 400 to 700 nanometers, and therefore the smallest thing that you can see under a light microscope would be about 200 nanometers, which is half the lower limit of the wavelength of visible light. And viruses will practically not be seen because they are smaller than that size. They are obligate intracellular pathogens, as I've said. So therefore, the difference from bacteria, that unlike bacteria which can grow on artificial media, and you might have seen some of those, or you might have been told that you can grow them on various artificial media, solid media and liquid media. You can't grow viruses in media in the absence of cells. So you have to have a system in which cells are present. I've already told you the other differences. And this depicts the two types of viruses which I mentioned, the enveloped ones and the non-enveloped ones. The first picture is of the non-enveloped one, in which you have a nucleic acid which is surrounded by a protein capsid, which, as you can see, is a very tight structure. And that property actually gives a huge advantage to these non-enveloped viruses, in the sense that they can survive much better in the environment, because the envelope, as I said, is primarily lipid, and it is quite susceptible to environmental factors, and therefore mostly enveloped viruses do not survive too long in the environment. You would have heard about the Karuna Virus and read about it, that it survives only very briefly in the environment, maybe a few hours, or if there's some surface on which it can find a foothold, then maybe a little longer, but not for too long. Whereas non-enveloped viruses survive even up to 6 months in the environment, in water, etc., which is why polio has been difficult to eradicate, because this polio virus, and in fact most of the viruses which infect via the gastrointestinal route, which means the viruses that cause acute gastroenteritis, you will find that all these viruses are non-enveloped viruses, because they have to enter via the fecal-oral route, and they have to survive in the stomach, in the acid, etc., before they can infect the gastrointestinal tract and then move on from there. So they have to be quite sturdy for that, and therefore they also survive better in the environment. So whether it be hepatitis A and E, which are transmitted through water, or it is the polio viruses and other enteroviruses, or the viruses that cause gastroenteritis like Rotor Virus, they all are non-enveloped viruses. So when we classify viruses, the first basic element of the classification is whether they are DNA viruses or RNA viruses. And then we go on to categorization, and you will find these tables in the books. I'm not going to go into those tables in detail. You will find that the DNA viruses can be either double-stranded DNA or single-stranded DNA. As far as DNA viruses go, practically all the families of DNA viruses have double-stranded DNA, and the exception is parvoviruses. So parvoviridae, you'll hear a lot of new names, so don't get intimidated by them, because you will learn about them over a period of time. And as you keep hearing those names, they'll start sounding familiar. So this is one of those classic MCQ scenarios where you are asked of the following, which DNA virus has a single-stranded, and there's only one family there, which is parvoviridae, basically. And the opposite holds true for RNA viruses. Again, practically all RNA viruses are single-stranded, except for the Rheoviridae or the rotavirus, which is double-stranded, which has a double-stranded RNA. Okay, so when we talk of single-stranded RNA viruses, obviously if they have one strand, that single strand could either be a positive sense strand or a negative sense strand. Therefore, single-stranded RNA viruses then again are further classified into positive sense or negative sense. What do we mean by positive sense? It means that you know that from DNA you make mRNA, and that's how, you know, the basic dictum that from DNA you get mRNA, from mRNA you get proteins or amino acids. And if you keep that in mind, then that means a positive sense RNA can directly act as mRNA. Whereas a negative sense RNA virus has to first get converted into a positive sense RNA virus, positive sense nucleic acid RNA, which means that it has to, so because you're starting from a negative sense RNA and you are moving on to a positive sense RNA, you need an RNA dependent RNA polymerase. And there is no enzyme in the host cell which is capable of doing that, because human cells don't require conversion from RNA to RNA. So you don't have that enzyme in the host cell, and therefore the viruses which are negative sense single-stranded RNA viruses would have to carry their own RNA-dependent RNA polymerase. And so they get certain properties which will be discussed in viral genetics. RNA-dependent RNA polymerase is an enzyme with a very poor proofreading activity. It's an enzyme which makes mistakes while it transcribes the positive strand from the negative one. And these mistakes actually help the virus because it leads to mutations. And these mutations help the virus to escape immune responses, vaccine responses, and it often gives the virus, by natural selection, a kind of survival advantage in some situations. Other than that, you also have DNA viruses as well as RNA viruses which have circular genomes. One of the classic ones is the human papilloma virus, which is a DNA virus which has a circular genome, and then there is another one which is the Hepatitis B virus, which is again a DNA virus which has a circular genome. Now, what are the various proteins and enzymes and the protein components, let us say, of the virus? We've talked about the nucleo component of the nucleocapsid, that is the genetic component, but the nucleic acid is very closely bound to the proteins which surround it. And these proteins, as I said, the capsid is made up of units called capsomeres, and these are very tightly packed. So based on the arrangement of the capsomeres, you can divide viruses into icosahedral or helical. I thought I had a picture of the icosahedron, but I don't seem to see it here. You'll find it in the book. Please take a look at the icosahedron. Okay, let me just show you. This naked nucleocapsid virus, the first picture that you see, and even inside the second one, that is the shape of an icosahedron. An icosahedron has 20 surfaces and 12 vertices, and it's a very beautifully packed structure. It's a very compact structure. So this is one. The other symmetry is the icosahedral symmetry, the helical symmetry, where you know that nucleic acid is helical in its shape, and therefore in this the proteins are comparatively loosely bound around that helix. So the helix of the nucleic acid with proteins surrounding them in a comparatively looser manner gives you the helical symmetry. So all these things are ways of classifying viruses: enveloped, non-enveloped, icosahedral, helical symmetry, RNA, DNA, single-stranded, double-stranded, etc. Linear genome, circular genome, all these things are used to classify viruses, apart from the actual sequence of their nucleic acid. Now, the proteins of the viruses are also this is a term you'll be coming across fairly often as we go on with virology, and these proteins are either structural proteins or they are non-structural proteins. When we say that they are structural proteins, it means that these proteins are a part of the composition of the virus itself, and they are a part of the physical structure of the virus. When we speak of non-structural proteins, these are proteins which are carried as the message in the genome, and when the virus goes into a host cell and starts replicating, these non-structural proteins are produced from the genetic code, and They are basically enzymes which are important in the replication of the virus, and there are a number of enzymes which are required for replication. So the viruses carry that message in their genome, and these non-structural proteins are not physically present in the virus usually, and they get expressed when they enter a host cell. So you will find, some of you might be familiar with the diagnosis of dengue. You may have had the test at some point of time. There's a test called the Ns1 Antigen Test for dengue, where you detect the antigen in the first 5 days of illness, and after that the antibody comes up and you can detect the IgM antibody. But this NS stands for non-structural. It's one of the non-structural proteins of the dengue virus which we are detecting. To come to the more important enzymes, I've already mentioned to you that in RNA viruses there is an RNA-dependent RNA polymerase, which is one enzyme which is not there in the host cell, and therefore the virus has to carry this enzyme with it. For DNA viruses, for the replication of DNA viruses or for making copies of DNA, there is the DNA-dependent DNA polymerase. This could be either cellular or viral. Because it is there in the host cell, the virus can use that enzyme, or some of the less efficient, more bulky, large viruses like the pox viruses will carry their own DNA-dependent DNA polymerase, and therefore pox viruses are larger than all other viruses. They are the largest viruses. In fact, the molluscum contagiosum virus, which is probably the largest virus, is almost there at the cusp of being visible under a light microscope. So this is because these viruses have to carry all these enzymes, etc. And it's a kind of redundant thing, but they do that. And then there are a couple of viruses which have RNA-dependent DNA polymerase or reverse transcriptase. It's not usually there. The classic one is HIV, human immunodeficiency virus, and also the Hepatitis B virus carries reverse transcriptase. These viruses, this reverse transcriptase is sometimes useful in the integration of the viral genome into the host cell. And they also have certain other enzymes like integrases which help with that. But other than that, there's the helicase, which allows the unwinding of the helical nucleic acid for replication purposes, and there are various proteases which are present, which are important in various steps of the life cycle of the virus. Now a lot of names over the next couple of slides, but don't worry too much about them. You'll remember them by and by. These are the DNA virus families. When we say family, actually I think the most common category of classification that we will be talking about would be the family. And we always end, and I'm sorry, there's a little mistake here. When we write the name of the family, it has to be in italics, and it always ends with Viridae, Viridae. So that is how the families are named. And as you know, in classification, just like you and I have to have names, the intent of any classification is to give names to various objects or forms of life, and that is why viruses have been named the way they have been. And within the family, you will find there are a number of different viruses. Some of them fall under other subgroups within the family, and they don't actually have a species name like bacteria do, or like fungi do, or like parasites do. The common name of the virus, if we say vaccinia virus or molluscum contagiosum virus or herpes simplex virus or human cytomegalovirus, that is considered equivalent to the species name. So each of these is equivalent to a species, although we don't usually use the term species here. Now within the family there will be fairly diverse viruses, but they'll usually have a common type of nucleic acid. It will have similar replication within that group, within that family. And its host cells or its target cells may be different. Viruses require receptors for their entry into a cell, and the virus can only enter a cell for which there is a receptor, on which there is a receptor for it. So you will find that there is a kind of host specificity as well as a specificity of the target cell which it can infect in the body. So whether, if it infects humans, it doesn't mean it infects all the cells in the human body. As you know, for example, the most prominent example right now is the Karuna Virus. You know that it will infect the respiratory epithelial cells because it has the receptors for that are present on the respiratory epithelial cells, which are the ACE2 receptors. So similarly, each virus has its own receptor and it has its own protein on the surface which allows it to bind to that receptor for entry. There can be other non-specific mechanisms of entry also, like endocytosis and so on, but some amount of specificity is imparted by the receptors. So I'm not going into the details of these families, but we have pox viruses, the Poxviridae family, and you must be knowing that smallpox, which has been eradicated, was a very prominent member of this family, which was eradicated by human intervention using vaccines, etc. There are still some other members which cause infection from time to time. The herpes viridae are a very important group of viruses. You have the herpes simplex virus 1 and 2. Apart from causing vesicular lesions in the skin, the importance of this group of viruses, the herpes viridae family per se, is that most of them can remain latent in the human body if you've got infected once. So if somebody gets infected with the herpes simplex virus, these viruses tend to remain in the ganglia for life. So it will be the trigeminal ganglia or it will be the sacral ganglia, but depending on whether it is 1 or 2, because 2 is usually spread by the genital route, although not always. And similarly, HSV1 is usually in the upper part, around the mouth and so on. So it's present in the trigeminal ganglia. So these viruses, all these viruses can remain latent in the body, and they can, in situations where there's stress or there's an immunocompromised state or long illness, sometimes just a cold, a low temperature, they can get triggered and they travel down the nerves and again start causing vesicles, etc. So if during your exams or especially in winter, you might find little vesicles around your mouth, that is possibly HSV1, which has popped up due to your stress. Similarly, human cytomegalovirus can remain latent in the body, and this is a very important virus of transplant infections, because when you give immunosuppressants to people, they can get these viruses, latent viruses, which can remain latent in the body for life, can get activated and cause infections in these people who have an immunocompromised state, either HIV infection or transplants where you have given them immunosuppressants, or malignancies, especially hematological malignancies, etc. Varicella zoster virus. I'm just trying to familiarize you with these families so that later on when you come across them, they don't look as intimidating as they possibly look now. The varicella zoster virus basically causes chickenpox in children, but later on it causes a condition called zoster, which is a very painful infection which travels down the nerve and infects a single dermatome. You know dermatome. You just finished with anatomy. So just a single dermatome with vesicles, and that too, and pain, nerve pain would indicate zoster, and this is a reactivation of the virus, latent virus in the elderly, especially when they are debilitated or they have some illnesses, chronic illnesses, etc. Or if they are immunocompromised. And then we have the Epstein-Barr virus, which is another one which can remain latent in the lymphocytes and get activated. In fact, it has been associated with certain lymphomas and so on and so. So these are some of the important members of herpes viridae. The next family is the Adenoviridae. The human adenovirus causes basically respiratory infections, pharyngoconjunctival fever. It also causes, a couple of them also cause gastrointestinal infections. A couple of these serotypes. Then you have the parvoviridae, and I mentioned parvovirus, the only single-stranded DNA virus family. And this parvovirus is basically, it leads to aplastic crisis in people who have aplastic anemia, and sometimes some childhood infections, etc., which are not that serious. The next family is papillomaviridae, which is an extremely important family because the human papilloma virus is one of the oncogenic viruses. It causes cancers, and especially one of the important cancers in women, the cancer of the uterine cervix. And it is preventable, this infection, because there's a vaccine now available. Then you have the, but it also causes some other cancers like anal cancers, etc., which are there in men. Oral cancers are also associated, some of them, the non-tobacco oral cancers. Then you have the family Poliomaviridae, which is, again, they are a little less common, but sometimes these viruses also get activated, just like the cytomegalovirus or the Epstein-Barr virus, in situations where people are immunocompromised, transplant recipients especially. And then, of course, you have a very important family, the last one in the DNA virus is the Hepadnaviridae, in which there's the Hepatitis B virus, an extremely important virus, especially for medical professionals. I expect that all of you would already be immunized against hepatitis B, but as you move to the clinics, I think you must make sure that you are immune and that you've been immunized. If you're not, then please get your hepatitis B immunization, because although most people will clear this infection if they get it as adults, at your age, about 25% might develop, become carriers of hepatitis B if you get the infection. So as you grow older, the chances of becoming a carrier are less. In neonates, it's almost 90% if they get this infection from their mother. So that's another reason to get immunized, especially for the girls. So I think this is a very important virus, and you have a good vaccine, so there's no reason why one should face the risk of this. And as healthcare professionals, because we are handling people's bodies and their body fluids and their blood, we have to be careful with this, because any small microabrasion and you could get any of these, these bloodborne viruses like hepatitis B, hepatitis C, very rarely HIV, because it's less transmissible. So the RNA viruses, the next group of viruses, and you have a long list here. Just to make you familiar, it's like a roll call. Picornaviridae, picoRNA, small RNA viruses, Picornaviridae, include the enteroviruses, and the most important enterovirus is the poliovirus. We are in the process of trying to eradicate it with substantial success, and a huge program in India, and I think so that's one of the sort of success stories, but then it keeps resurging in some countries here and there, and still causes us worry. It causes acute flaccid paralysis, although 99% of people will just get an asymptomatic infection. And that is another property of viruses. They can often cause asymptomatic infections, which you again, in the context of coronaviruses, have been hearing a lot about. A lot of respiratory infections, a lot of these enteroviral infections can be totally asymptomatic, and you may just get the infection, not know, or get a very mild infection and seroconvert. Seroconvert meaning you will develop antibodies or develop an immune response to it, without even getting clinical features. And that sort of thing happens in a number of viruses, and especially true of Karuna. Hepatitis A virus is another very important member of Karuna, but it's in a sort of different subgroup within the family. And then there are the Rhino viruses, which cause common cold, and there are a large variety of them, therefore you can keep getting common cold often. The next important family is the Orthomyxoviridae. This family basically has the influenza viruses. And then the Paramyxoviridae, which is again a very important family, because it not only has the para-influenza viruses 1, 2, 3 and 4, but it also has some very important childhood viruses like measles and mumps virus, for both of which there are vaccines, good vaccines available, and they are part of the immunization program, childhood immunization. But you also have some new viruses like the human metapneumovirus, and one which is not listed here, but it's become very important because it pops up from time to time and creates a lot of problem because it causes CNS infection, and that is the Nipah virus. Also falls in the family Paramyxoviridae. But a more common member of Paramyxoviridae is also respiratory virus. Measles spreads by the respiratory route, mumps, parainfluenza. These viruses spread by the respiratory route, Nipah. A very important member is the respiratory syncytial virus, or RSV, which is a member of the Paramyxoviridae. And respiratory syncytial virus is a very common childhood infection, especially in winters, where it causes bronchiolitis. And these people have to get admitted for bronchodilation, etc. So these children, it's an important illness of childhood. And then the current top of the charts, Karuna family. Well, we've listed just SARS and MERS here, but there are a lot of coronaviruses which have been there from ages. This is not the first coronavirus which has come up. And even before the first SARS virus, this is of course the current one is called SARS coronavirus 2. Even before that, there are a number of coronaviruses which are already there, which are also in circulation, especially in children. But in our developing countries, they remain in circulation in the respiratory infection season, and possibly might be affording us some amount of cross protection, which might be useful, at least in preventing severe disease, because we have been hearing a lot about why it is less severe in Indians and in the South Asians and so on, in our part of the country, why less deaths. I don't know. We still don't know a lot of things. But this could be a reason, apart from the fact that we have a younger population, which is in any case not getting as severe infection, which doesn't mean that you guys are going to, the moment the town is open, we're going to go gallivanting around. That is not the intent. Kindly keep in mind that you have elderly at home, elderly people, and you're moving around without taking safety precautions like masks and social distancing or physical distancing, both of which are equally important. Just masks are not enough, just physical distancing is not enough. Both have to be used. So if you are not careful as youngsters, thinking that your risk is lower, you are actually compromising older people in your family or in your surroundings. So just remember that. I want to give this message today because this is the first class in virology and we are in the middle of a pandemic. People are dying because of it, and I think it's extremely important that you understand your own responsibilities and roles in preventing the transmission of this infection. I think that's enough about coronavirus for today. You've probably been hearing this message for a long time, but there's no harm in reiterating it. And then we have the Rheoviridae, the human rotaviruses. The rotavirus is the most important cause of diarrhea in childhood, especially in children below 1 year, infants. but it can cause infection in young children less than 2 years, 3 years. So, and it can cause repeated infection. This is the most important cause, and in fact, once we came to know and once we learned that a lot of infection in neonates and infants is due to rotavirus and not due to a bacterial cause, the use of antibiotics, etc., has been limited, which has allowed us to save lives, because all you need to do in these babies is to replenish their fluids. And for that, you can use oral rehydration solution. If you use antibiotics, you are actually compromising their gut flora, their normal flora, and that can lead to more complications. And therefore, since we realize now that a large majority of childhood diarrheas, and especially neonatal and infant diarrheas, are due to rotaviruses, they are viral in nature, and you don't really need to do anything more than just replenishing their fluids. If they are too dehydrated, you can give them IV fluids, but mostly they should settle down over a short period of time with just oral fluid replenishment based on the WHO formula of the ORS. We also have other diarrhea causing viruses in this family which are less common, but they are there. And in adult diarrheas, Galiciviridae is an important family, the noroviruses, the sapoviruses, etc. And there is also another family called the Astroviridae, which are important causes of gastroenteritis, acute gastroenteritis. Then the next family is the Hepiviridae, which contains the Hepatitis E virus. So all these viruses, Rotor, Noro, human astroviruses, Kelly viruses, Hepatitis E virus, and Hepatitis A virus that went before, all these are spread through, they are waterborne or foodborne, etc. And all these viruses are able to survive in the environment, and as I told you, they are non-enveloped viruses. That's why they are able to survive better and get transmitted through water. Even tolerate low levels of chlorine, etc. Then you have, and they cause outbreaks also because of that. Then you have the family Rhabdoviridae, which has the Rabies virus, which is extremely important in South Asia and in India, because we possibly contribute large number of rabies, the largest proportion of rabies cases at this point of time. And you know about rabies, how it spread through dogs, dog bites, etc. Another important family of RNA viruses is the Flaviviridae family. And again, we are in the middle of the season for one of these, and that is dengue. The dengue virus season is the monsoon. And so we are concerned, and there again, as responsible members of society and responsible citizens, there are cases happening in your hostel. The authorities can do only so much about it, which they will do. You can do a lot about it, because the mosquito which spreads dengue is it doesn't move very far. Its flight range is somewhere around 100 meters or so. So if you have water collections in or around your rooms, just keep an eye out for those and just empty them out. Just keep those areas dry. If you want, you can add a bit of just get some kerosene from the mess or somewhere and just pour a little bit of kerosene on it, but it doesn't matter. I mean, or you can at least just do a mosquito spray or whatever. But the thing is, keep it dry. Even a small container, even a teaspoonful of water, if it is left outside in this weather, can permit breeding of Aedes. And it'll be in your immediate surroundings. And who's going to empty out that little paper cup that was thrown away just outside your hostel room? Maybe they'll clean it up, but maybe it just sort of sits in one of the bushes there and collects a few spoons of water, and the mosquitoes start breeding there. Just keep an eye out for it. So that's the dengue virus, which basically causes fever or even asymptomatic infection, but in few people it can lead to dengue hemorrhagic fever and shock and severe dengue. You also have the Japanese encephalitis virus in the Flaviviridae family, again transmitted by mosquitoes, but not by the Aedes mosquito. JE is transmitted by the Culex mosquito in it. And you have some of the yellow fever virus, again mosquito borne, and then some of these tick borne viruses like Casanova forest disease, which are localized to certain parts of India, especially down south, but is moving up a little now and moving to surrounding states. And the most important member of flaviviridae right now, apart from perhaps dengue and JE, is hepatitis C virus. Hepatitis C virus is a blood-borne hepatitis virus, just like the hepatitis B virus. It's also a blood-borne virus, just like HIV in some ways. But the good thing about hepatitis C is that now you have excellent drugs to treat it, and you can actually eradicate the virus by giving treatment. And therefore, it's important that if this infection happens, it gets diagnosed quickly and it gets treated. There is no vaccine for this, unlike hepatitis B, and it is also a risk for healthcare professionals. So, but the good news is that there is good treatment now available, short treatment, maybe a few months, and you should be able to clear the virus. Then there is the Togavirid family, and this has a very important virus, the rubella virus, which causes congenital infections transmitted from mothers to the babies. The other important virus which causes congenital infections is the Rna virus, which is the cytomegalovirus which we talked about. But the problem with rubella is that unlike cytomegalovirus, which causes some amount of congenital anomalies, the rubella virus, if it happens in the first trimester, is almost certain to cause to be transmitted to the fetus and cause anomalies. So even more than 80% of the babies would get infected, and more than 80% of those would get congenital anomalies if you don't prevent it, and the primary infection occurs in the first trimester of pregnancy, which is why women need to be immunized for rubella. They must be sure right at the start of pregnancy that they have IgG antibodies to the rubella virus, which indicate past infection or vaccination. And a certain amount of those IgG antibodies would make you protected against this infection during pregnancy. And then you have the Bunyaviridae, which are again rather exotic. Crimean Congo hemorrhagic fever, which we've seen in some parts of Gujarat and surrounding states, Maharashtra, etc. Some outbreaks from time to time, small outbreaks, but some spread is there in India of this virus. And it's a hemorrhagic fever again, and can be quite dangerous and fatal in a certain percentage of people. Hantan virus, which is a rodent-borne virus. So you see there are a lot of viruses which are transmitted by insects, which we call arboviruses, and a lot of them which are transmitted by rodents also, at least some of them by rodents also. Rna Viruses, not so important, can be transmitted, is also transmitted between mice, etc. Then you have the Filoviridae, the famous Ebola virus, or rather the infamous Ebola virus, and another member of the family called the Marburg virus, which have high case fatality rates and are transmitted quite efficiently through blood and body fluids. Fortunately for us, we don't see this virus in our part of the world. Finally, we have the Retroviridae family. As I said, it has reverse transcriptase and hence the name Retroviridae. And in this you have the HIV and the HTLV. So HIV 1 and 2, the human immunodeficiency virus, and the human T lymphotropic viruses, which are comparatively less common and less important in our population. So this is a long introduction to all the viruses. Let's move on from here. Just a few pictures. The pox virus, Poxviridae, you see the shape. This is the helical symmetry, because you have these proteins which are loosely bound around the helical shape of the nucleic acid. The circular viruses, circular genome, the hepatitis B virus below that. You see a lot of projections on the surface of these viruses, the orange colored ones, and even on the herpes viridae. These projections are basically surface glycoproteins which jut out of the envelope in enveloped viruses, and they you can see this in the electron microscopy picture, the transmission electron microscopy picture of the adenovirus on the right. These are the structures which jut out and they allow the virus to bind to the host cell. They usually have the receptor of the virus for the host cell on them. Here are some of the RNA viruses. Again, you see some of these with these structures on the surface. HIV has these. It has what is called GP120, which is projecting out and binds to the CD4 receptor. You have others also with similar situations. You see a nice electron microscope picture of the coronavirus. This you can see why it is called the coronavirus, because it has this kind of corona crown around it, which is again due to these spikes which protrude from the surface of the virus and allow it to bind to the ACE receptors in the host cell. The Rhabdo virus, very typically bullet-shaped cylindrical virus. Rabies, the one on the extreme right. Replication, I'm not going to go into too much detail. I'll just take maybe 5 minutes on it just to give you an idea. See, the basic thing is the virus has to bind to its receptor. After that, once it binds to the receptor, then the nucleic acid has to enter the cell. Inside the cell, the nucleic acid is then either in the nucleus or in the cytoplasm. This will replicate. The viruses which carry their own enzymes, like the pox viruses, don't need to go to the nucleus because they have their own DNA-dependent DNA polymerase, so they'll replicate in the cytoplasm. So those which need the enzymes from the nucleus will enter the nucleus. So usually there are some principles behind this replication. In any case, once they replicate, they make more nucleic acid, and some of this gets expressed on the surface of the cell, especially the surface proteins. Proteins tend to get expressed on the surface of the cell. And as the viruses which are enveloped, when they exit the cell, they do so by budding. And as they bud out, they carry those proteins from the surface of the cell to form their outermost layer or envelope. So you can detect the presence of these antigens on the surface of cells by various methods like immunofluorescence, etc. And it has implications for diagnosis. The other mechanism by which viruses exit the cell is by lysis of the host cell, which means that the host cell gets destroyed and these viruses come out by lysing the host cell, and these would be mostly the non-enveloped viruses. The enveloped viruses, because they have a lipid membrane, this membrane can fuse with the membrane of the host cell, and it will allow a more subtle entry into the cell and exit from the cell. Whereas the non-enveloped ones just have to come out by lysing the cell. So these are some of the principles of replication. And we are just showing you here this picture of herpes simplex virus replication. There it is phased out. Like I told you, the regulatory or non-structural proteins, there are these immediate early proteins which are first manufactured. Once the virus enters the host cell and shuts down the machinery of the host cell, hijacks it and starts using it for its own replication. That is what really happens. And in that process, the immediate early ones are made first, and this kind of phasing happens. So these are used for they control the replication of the subsequent genes, and then come the enzymes, which are the early proteins. And these enzymes are needed for viral replication. You can actually, there's this enzyme called thymidine kinase in herpes simplex virus, which is extremely important for its replication, and we have a wonderful drug which targets it, which is safe and can treat herpes infections. Herpes simplex virus infections, particularly CNS infections, it can lead to encephalitis, so especially HSV1. And this drug is extremely important in saving lives in those situations where people get severe herpes infection like encephalitis. And these drugs target the early proteins, so the thymidine kinase specifically. And then you have the late proteins, which are the structural proteins, which ultimately form the various components of physical components of the virus before they exit the cell. So these are some of the steps in the replication, and different viruses have slightly different mechanisms of replication. I'm not going into too much detail. I don't think it's that important. Where it is important, when we talk of the individual viruses, we'll let you know. Like for example, the influenza virus has a hemagglutinin which binds to this, which is there on the, which is the antigen on the virus which binds to the sialic acid receptors in the respiratory membrane. So each one has its own, and these are all important because the HA, you have a vaccine for influenza based on the hemagglutinin, which changes every year because the hemagglutinin keeps subtly changing by point mutations every year. HIV, of course, the classic, binds to CD4 receptors and some of the co-receptors on the surface of the cell, the lymphocyte, and enters the cell by binding through its GP120 viral receptor, and goes inside, uses reverse transcriptase for its replication, and then comes out by budding eventually, because it is also an enveloped virus. We have already talked of the clinical presentation, so I don't think I need to give you these examples. We've already talked of the kind of infections. You have respiratory infections. I've already mentioned the viruses which cause respiratory infections, like the influenza virus, parainfluenza viruses, respiratory syncytial virus, human metapneumovirus, Karuna Viruses, Rna Viruses, which cause common cold, the last one. So they can cause either upper or lower respiratory tract infection, even pneumonias, etc. in severe cases. You have the viruses which causes exanthematous disease, especially in children, the measles virus, which, as I told you, belongs to the family Paramyxoviridae, the rubella virus, which causes German measles in children, but is more important in transmission from mothers to their babies as a cause of, preventable cause of congenital infection and congenital anomalies, because you have an excellent measles, mumps and rubella vaccine, which is a live vaccine given in childhood. And then of course you have chickenpox, varicella, which have different patterns of rash and so on. For example, in measles, the rash is basically macular, which is just slightly raised, whereas in chickenpox, it would be a vesicular rash, just like in herpes simplex virus, there's a vesicular rash. So it forms vesicles which eventually rupture and settle down, but you know, some amount of scarring can happen and so on. So these are the way, and then you have the petechial rash, which is there in hemorrhagic fevers. So there are all these examples of rash with fever, many important in childhood, others important later also. You have gastroenteritis. I've already mentioned the viruses which cause that, diarrhea, vomiting, etc. Rotor virus, especially in children less than 2 years, it's extremely important. Adenovirus, serotypes 40 and 41 I mentioned. Noroviruses, which cause adult viral diarrheas, acute diarrheas, and astroviruses, etc., which also cause diarrhea, gastroenteritis. And then you have the hepatitis viruses, A, B, C, D, and E. As I told you, hepatitis A and hepatitis E are both transmitted by the fecal-oral route, and they are non-enveloped viruses. And then you have the blood-borne hepatitis viruses, hepatitis B and hepatitis C. Hepatitis D is a satellite virus. It does not exist on its own. It infects only those people in which hepatitis B is already replicating, because it takes some of its outer coverings from those of the hepatitis B virus. So this sort of causes either superinfection or co-infection with hepatitis B, and doesn't exist on its own, which is why it's like a satellite virus, like some of the plant viruses. The acute CNS infections, encephalitis, meningitis, acute flaccid paralysis as in polio and some of the other enteroviruses, acute encephalitis, which is caused by Japanese encephalitis virus in our country, extremely important, especially in the eastern UP belt, where it has become endemic and causes cases every year in surrounding areas. Again, there's a vaccine for it, so it can be prevented, and it's spread by mosquitoes, the Culex mosquitoes. But then there are other causes of sporadic encephalitis, like the herpes simplex virus, which I told you is eminently treatable by thymidine kinase inhibitors like acyclovir, which is a drug for it. Rabies also causes encephalitis, central nervous system involvement. So that also gets classified here. And mumps can be a less common but important cause of encephalitis also. Aseptic meningitis, which is different from pyogenic meningitis, which bacteria cause, is also caused by many of the enteroviruses. And this is different because it doesn't have too many pus cells. You'll find a small number of lymphocytes, usually less than 100, whereas in bacterial meningitis, acute pyogenic meningitis, due to Neisseria and Haemophilus and pneumococcus, you will find a lot of pus cells, basically neutrophilic response rather than a small lymphocytic response. That's how you differentiate it. Apart from looking at the picture of the CSF in terms of the biochemical picture, which means that the bacterial infections, actually the bacteria use up the sugar, so there is a low sugar and a high protein in bacterial acute pyogenic encephalitis, whereas in viral you don't see those things. So that brings us to the end of this long introduction to, I think we took about an hour, so we are pretty much on time. What I'll do is I'll give you a break of 5 minutes and take a break of 5 minutes to just relax a bit, and we come back again and we discuss the laboratory diagnosis. What are the tests that we do, what samples do we collect, how we collect them, how we transport them, what are the various modalities of tests, when they should be done, etc. over the next, say, 40-45 minutes after a 5-minute break. So see you guys again soon.